Hi, good evening, all the participants. Thank you for joining us this evening for a talk under the CVJ talk series. Professor Pawan Dhar I'm introducing the speaker. Professor Pawan Dhar is the Executive Director of the CVJ Centre for Synthetic Biology and Biomanufacturing, which is associated with Kochi University of Science and Technology, and a pioneering figure in shaping India's synthetic biology ecosystem. Professor Dhar brings together academic leadership, institution building, and entrepreneurial mentorship, backed by more than 33 years of international experience across India, Japan, Singapore, Germany. Uh, and before joining the CVJ Centre, he headed the School of Biotechnology at Jawaharlal Nehru University, New Delhi, and his scientific contributions span across synthetic biology, genome engineering, and exploration of dark genome, the largely, uncharacterized regions of DNA beyond conventional protein coding sequences. So his work in this emerging frontier has led to the world's first demonstration of drug discovery drug discovery pipeline originating from intergenic regions of DNA, opening new possibilities for the therapeutic discovery from previously overlooked genomic spaces. And today, Professor Pawan Dhar will be giving an introductory talk on synthetic biology and its possibilities. This talk marks the second lecture in the CVJ Centre talk series, and it is a pleasure to invite Professor Pawan Dhar to take the floor. Thank you, Pawan. Please.
Thank you very much, Dr. Unikrishnan, for your very generous introduction. That increases my responsibility and expectation of the audience, I guess. So this is the second talk in the series, as you just heard. The whole idea is to bring audience up to the speed of the technology that is currently being practiced so that we can explore jointly the journey forward. And given that the technology is rapidly evolving, we also have to evolve our concepts and our hands-on experience at the same pace, not to be left behind. So today, I brought a bunch of slides to initiate you into this thought process. My introduction into synthetic biology happened. I was fortunate to be there when the field was announced at MIT Boston back in June 2004, when the first synthetic biology conference was organized. And then with Professor Ron Weiss from Princeton University, we started a journal called Systems in Synthetic Biology with Springer. And then, things kept moving. What has happened is over a period of time, people have got different versions of synthetic biology, and different definitions and variations have come up. So I want to bring you back to the very basics where the whole field was started and why this was started. What was the reason to have this new terminology when you already have genetic engineering? Well, we had genetic engineering, but genetic engineering itself is not an appropriate term. It would be rather correct to say recombinant DNA technology, because we never engineered genetics. This is the first time that engineering principles are coming into picture and help us build the systems. So what do we mean by that? How is genetic engineering different than engineering biology that we are talking right now? So in the following slides, I hope it'll be very clear as to what we are talking. You see, whenever technology came up in the market, it has rewritten something, whether it was printing, it was computer, or it was internet. Something has been rewritten substantially. And then you have a new market, new mindset, new users, new early adopters and late adopters, and new way of negotiating with the technology, new way of adopting the technology. Likewise, in this sequence, what has now come up is the ability to rewrite life. When you say life, of course, we have not touched the life operating system. However, the biological operating system that we are talking of in terms of the genome sequence is what we are referring to the rewrite. Um, we are the first generation of humans who can rewrite a new DNA code. Maybe it was not possible 10 years back if you ask somebody, or maybe 20 years back. We have gone to the level of writing primers, which are small DNA sequences. However, the writing technology has phenomenally, gone ahead with increasing the length of DNA and decreasing the errors and making it more affordable. Thereby, the old style of recombinant DNA technology, wherein you lift the sequence and paste them in a vector, they are slowly becoming old and they're slowly getting out of the fashion. So, what is coming up and what is already there is the ability to type the DNA sequence in the computer, email the DNA sequence to the DNA synthesis company, and they give you the vector of your choice. So 1 year of effort goes down to maybe a few weeks. Um, so that is the speed that has been achieved because of technology. Biology, in a big way, has moved from reading and writing and editing. When you say writing, it is also editing because you lift a piece of DNA, you replace it with something. You're not writing the whole story, you're writing one paragraph of it. But still, it is a phenomenal empowerment. So, by the end of this lecture, I hope you don't ask yourself what is synthetic biology. Rather, you ask yourself, can you write the future versions of the genome? Now, we're not talking of the definitions, we're talking about capabilities here. And when you write the future versions, we're talking the source code, which is a DNA sequence. We're talking the hardware, which is a cell. And we scientists have become programmers over a period of time. And the whole journey, if you look at the classic way of handling this, the entire journey is what we call as reductionism. Going back to the first principles of science. We have an organism. We want to explain what it is. How do you do that? You have to decompose the organism and then finally reach a level at which further decomposition is not possible, going from organisms to molecule. And this journey is called reductionism. You basically reduce the size of investigation. And how do you start? You start by looking at the patterns at the phenotypic level, at the external observation level. You look at the patterns, and then you ask yourself, well, they are looking very similar. What is the fundamental reason of this similarity? And then your journey of science begins. Now you dive deep into the second layer, which you call as gross anatomy. Here you see a bunch of bones and cartilages and blood vessels and nerves and other tissues coming in collaboration to boot up a human system. But at this level, you get a slightly more detailed view of human body. However, when you touch even one aspect of it, let's say soft tissue or the hard tissue, and you go deep, you see there's a new ecosystem emerging. And this is called histology. When you, for the first time, you place the tissues under microscope and you want to study them, you get a new view of biology, which was not visible when you look at the cover of the body. And you go to the next level, and then a new vista is unveiled. Now, you're no longer talking about the multiple cells. You're just talking the single cell using chemistry, using microscope, and using the other optics technology. You perfect the staining part, the visualization part, and then you figure out that there is a lot more in the cell than you ever imagined because it was looking very plain vanilla from outside. Now you have nucleus, you have cytoplasm, you have organelles, and so on. Now, when you zoom in further, for example, in the nucleus, you find that there are chromosomes. That's because this is the largest structure that's visible at 1500x magnification. So this is an affordable data collection. But when you look at the chromosomes, then you realize that it is nothing but a DNA sequence, which is coming in contact with the histone molecules. at least 8 to 10,000 times. It is coiling to make a chromosome and uncoiling to make chromatin. You get fascinated by these molecules, and then you ask yourself, have I finally reached an end point of my scientific pursuit? Well, that is what was perceived back then, 20s and 30s and 40s, when DNA got a superstar status and it was considered as a megastar. Everybody is chasing DNA sequence, and so many publications coming. For nearly 40 years, it has achieved that status. But then the RNA community caught up, the protein community caught up. So you have more of biochemistry now coming in, and transcriptome, proteome, those kind of concepts and data came up. And now DNA went back and lost its superstar status. And now it is being perceived more as a database of information. That's the beauty of science. But is DNA our last destination? No, because when you go beyond DNA, you see that it is nothing but chemistry. Chemistry happening in biology, and we call it biological chemistry or biochemistry. Now, when you look at the chemistry, remember what we started. We started from organisms in this reductionist journey in order to see the composition of molecules. So we are not yet touched the final frontier. But when you see the what is chemistry composed of, what are the enablers of chemistry, then you reach the level of atom. Here you become an atomic physicist. Now, atoms talk to each other and chemistry is born. Chemistry happens to be in biological systems and biochemistry is born. Biochemistry is enveloped and cells are born, and cells are talking to each other, tissues are born, and likewise, a highly complex eukaryote boots up. But when you reach this level, you realize one thing, and that's what makes you philosophical, that till now, I was able to see something, whether it was tissue, whether it was cell, whether it was molecule or organelle, I was able to see something. However, now I'm hitting the darkness because the light is not hitting anything. Why it is not hitting? Because the 99.99% is space. So which means that all the data that we collected till now, all the data that we call it science, is due to collision. So collision is the fundamental data gathering method because light collided with something and hit our retina or hit the lenses, and the image of a particular surface is born. And you capture it in the database and so on and so forth. But collision is the fundamental requirement for any scientific pursuit. Well, collision is fine, but also when it hits us, when the light rays hit us, it has to be compiled. So you have a physical data which comes to us because of the collision of light. And if there is no collision, we don't have the data. So now this collision is going through 5 compilers that we call senses. And then a chemical soup is made, or maybe an electrochemical soup is made, stored in different parts of the brain, and then you visualize or you realize there is something in front of me. Therefore, what it tells you is that nature presents itself in the form of physics. We understand ourselves, or we understand nature in the form of chemistry. And it is our compilers that enable this transformation. So different people have different levels of compilers, so different levels of data. But end of the day, if you take a final reductionist view of life, you go from pedigree, 28 symbols that we represent the family trees, then you go to anatomy and so on, finally end up in quantum physics. You're talking of possibilities. You're not talking of specific atoms here. You're not talking of the subatomic particles here. You're talking of possibilities giving rise to reality, which means if you have to see what is my final reductionist view of biology, then you have to go to the level of possibilities, and the compiler between possibilities and realities is what generates the data. So not to make it too philosophical, let's stay at the level of reductionism that we are aware that we are talking of the cells, and the cells are basically um, we investigate these cells and they make molecules. So this journey has been very successful. Now, when you have a lot of, let's say these molecules are nothing but genes and proteins and RNA. Now, when you have a lot of them, what do? You cannot physically keep a record of these molecules. You have to transfer the data to the databases. So now when you transfer the databases, this data to the databases, capture it, we are essentially talking of representing the data or modeling the data. Database itself is a model of reality, which we call as biology in this context. And when you represent the data, first it is a single molecule, then it is an interaction of molecule, and then this interaction grow into the pathways and so on. Well, these are two ways of doing science historically. So it so happened that nearly 2 decades back, 2.5 decades back actually, people asked, uh, can we add a third approach to science? Which means, can we construct something? Say, for example, today we are interacting through computers and mobile phones. This was not made by nature. This is made by humans. So now, can we bring that construction approach into biology? That's what was asked in this meeting. So what does it mean? Can we compose organisms? Can we compose mitochondria? Can we chemically synthesize ribosomes? Can we chemically synthesize the genome and the cell and so on? Construction approach in biology gave rise to a phenomenal understanding of the challenges and the opportunities. So the first one we know as reductionism. And so the first one is more popularly called with these names that you all know better than I do. But the second one, the representation part, is what we know as bioinformatics and systems biology. Bioinformatics, when you talk of single molecule study, and systems biology, when we talk of collection of molecules giving rise to certain behavior. And the third approach, which is a construction approach, is what we call as synthetic biology. So synthetic biology got a bad press in the beginning. Of course, it got a good press also, but people started thinking a lot of stuff around this. So many people prefer to call it biological engineering rather than synthetic biology. And early on, I remember in the discussions, people were suggesting that we should call this as constructive biology. Then there were objections. Well, is the rest of the biology destructive? So let's call it biosystems engineering. Then if you call systems, then the components, the parts are not taken care. So you've got to, include the parts also in the definition. So then somebody said, let's call it biological technology. Then what's biotechnology? So finally, the community settled with this insight that biological engineering is the appropriate name for synthetic biology. So you have papers that say biological engineering or publications that say synthetic biology. They mean the same thing. Okay, so just want to clarify this before moving further. So at the systems biology level, which is basically the foundation of the rules and standards you get for synthetic biology. This is where the journey began in Japan. They created a virtual red blood cell, and I was part of this project. We learned from there, and what you see on the screen is nothing but C++ code and ordinary differential equations. And some of them were stochastic equations, especially some of the gene expression events. But essentially, as a biologist, you have to program the cells. So when you click on any button, you're clicking, you're switching on the gene or switching off the gene. With that kind of capability, they made some phenomenal discoveries. So, this was in 1998, 1999. This first paper was published from a Japanese group, Professor Masuro Tomita, who came from Carnegie Mellon. And with Craig Venter, he published this paper. And people have gone further to the extent of saying, can we capture more details in eukaryotic cells? And can we create a virtual human skin? And this is, by the way, a $10 billion market. And if you can create a computational model of this human skin that can model the processes of wrinkles, that is the aging, the color, and hair growth, it's a phenomenal application waiting for you. So, this was a thought paper which was proposed at that time. Because we have a lot of data and a lot of standards, is it time to construct a periodic table in biology? If yes, then what would be the element of this bioperiodic table? I was in Japan at that time, and Journal of Biological Sciences, Journal of Bioscience from Bangalore, they were very generous to publish the paper because there's no proof of this. This is only a thought paper, and it was rejected by many journals. So they were very generous to accommodate this thought. The question is, we are now talking of ability to think like an engineer. As I said earlier, genetic engineering is definitely there, but we never engineered genetics. We are talking of here using engineering practices. So what do you mean by that? Well, there are dissimilarities between engineering and biology, for sure. We know this. Engineering systems are robust, and biological systems are linear and non-linear, and predominantly analog. And when you look at the cells, there are millions and millions of molecules and millions of transactions happening at the same time. There are dissimilarities, but there are also similarities. But in this case, what is important is to understand is that biological systems are noisy and unpredictable. So what do you mean by that? Noisy means that the expression levels go up and down very frequently. And unpredictable means that if you knock out a gene or if you knock in a gene, you do not know what's going to happen. Okay, so there's a bit of unpredictability around biology, all the modifications that you do. And people are talking of creating gene circuits, but it is not like the gene circuit of the engineering level, because over there you just set up the rules and the information pathways, and it will follow you. However, in biological circuits, the information has to go several layers down. Even if it has to go to the next level, that next level will come when the underground level 1 and underground level 2 is cleared. Then you go to the next level. Okay, as you can see in the screen. But there is a little more, little more than that, because it is not just the mRNA. It is also that enough mRNA should be there to trigger the next event, and there is a massive mRNA degradation. So for any sequence to go to the level of protein, any expression to the level of protein, and protein becoming a transcription factor for the next gene, it has to survive the cellular challenges in terms of the degradation, in terms of the availability. Okay, so it's not like hardwiring something in the motherboard and activating it. It is far more challenging than that. So to design well-behaved systems, one needs standards and rules of composition. Well, standards are something like this, that, a particular rail is fixed. Let's say some measurements are fixed, so you do not change them. You say, okay, whether it was good to have 5 feet 7 inch or 10 inch or 5 inch or whatever be the size doesn't matter here. What matters is that, are you keeping this constant or are you changing it? So you have broad gauge, you have meter gauge, you have narrow gauge, and then you have machines which are mapped to the width of the rails, and the technology is delivered accordingly, but something is fixed. In our case, do we have that at the moment? We'll come to that. So the second thing is rules of composition. Now that you have standards, in what order or in what sequence do you place the parts so that the whole system works. Say, for example, if you have to create a lac operon, or you have to create a metabolic pathway, you have to design a glycolysis and install glycolysis in a cell that does not have the glycolysis, then you got to see how many genes do I take? How many, well, every gene is under a regulation of a certain promoter and certain transcription factor and certain inducers. So which means now your problem becomes, enormous. It's not just 10 gene step or 10 enzyme. It is also the triggers of those enzymes, transcription factors. You have got 10 more players to handle. And you also have to regulate their expression. You have 20 more players to handle. Back then, we found that if we have to, this was done quite some time back, we found that if we have to install a glycolysis pathway, you have to handle 42 components in an E. coli. So it's non-linear because you've got to take care of the regulation part, not only the process, but also the regulation. So people have come up with this representation back then. I remember this is what was discussed in the first meeting of synthetic biology um, at MIT, that how about if we create 6,000 truth tables in yeast and 4,000 truth tables in E. coli? Can you compose E. coli? Can you compose yeast? Well, maybe conceptually, yes, but practically, no. Why? Because you're not talking of a linear system, you're talking of a non-linear system. And all the values that you see in the table, they're not fixed values. They are dependent on the context. So which means that that particular organism, that culture condition, that metabolic state, and the value holds true. And as you know, as you grow the organism, the environment keeps changing. So you cannot have a fixed environment. Therefore, the values do not make any sense. And that is why this particular initiative, which was considered as a very innovative approach, it was not really pursued because of the inability of, and the grand challenge is really to engineer biomolecules, circuits, and using construction standards, if we can do. Now, we're talking of standards, but we also know that the IEEE standards do not exist in biology simply because that is not a problem of organism, that's a human problem. So why should organism worry about the human standards? So what's the solution? The solution is that you decompose the biology, the system one part at a time, which is what we have done in the traditional settings. And now we are talking of composing the system one part at a time, but it has to be a controlled construction. And this controlled construction means that a responsible innovation, remember now you're taking the role of the creator. You're creating something for the first time, and it has to be exactly as the specifications are written. It cannot be that we started with the E. coli and we ended up with Ebola virus. We should not be, we should be very careful about, what we are constructing. Of course, there are checks and balances and safety parameters at every step, so nothing to worry. But at the same time, one has to be very clear as to that we are now constructing a nonlinear system, which is non-trivial. But if we are able to do that and we are able to compose biology, as you can see, the payoff is enormous. In the next 2 decades, we are talking about 2 to 3 trillion market, not billion market. And that is the way the biology has evolved. The practices have evolved. Now you can see the parallel between synthetic biology or engineering biological systems and our traditional practices. So initially, when we were in early 60s, there were individual labs. People used to work in small labs. Then came a concept of CIF, that you have these central instrumentation facilities. These are the core instrumentation facilities in an institution. So instead of spreading it around, you basically collect all of them in one place and everybody uses them. Then came the concept of CROs. You have the contract research organization. So you run out of the budget and you ask them, okay, can you do this for me? And then came the concept of bioincubators in 2001, wherein the startup companies are basically achieving a certain objective that has a commercial endpoint. But the latest practice, and why I'm putting this slide here is because when you talk of engineering biology, you're talking of speed and accuracy and standards. You're talking of affordability. And that is where biofoundries are now established, and I'm sure you know about this Government of India initiative of creating biofoundries across the nation and regional biofoundries. What will they do? They'll basically build the standard not only in terms of the data sheets of the parts, but also in terms of the processes that you would follow. If you have to do a gene expression measurement, or if you have to do a recombinant DNA technique, what would be the best way to go forward? What's the recommendation? What's the boundary condition? What's the troubleshooting table? All of this builds into standards, and standards have essentially the data sheet, which means that it's not only talking about the behavior of the part, but also its best behavior in a certain context. And then comes automation and robotics, wherein the human errors are minimized and robots are taking over all the repetition job. And then the high throughput screening, because you end up with thousands of designs and you don't know what to do, which one is correct. And the machines help you in selecting the right designs and say, okay, I'll put some constraint, you do random mutagenesis, I'll do the directed evolution by putting some constraints and screening a colony of cells so that you quickly reach your design, which may be embedded in thousands of innovations. And this again has to be driven by AI, because you need the help of computation to basically help you arrive at a certain design which is stable and which can be also inherited, and it can be stable across the generations. So the biggest concern, there was a particular survey recently by Accenture. They said we have a severe shortage of skilled labor, skilled people. The need of the future is bioengineers who can code, fermentation experts who can use AI tools, and technicians who are programmed for industrial manufacturing. Well, this has already started way back, when bioinformatics was launched as a field mid 90s. Many of us had to go through programming classes, even after doing PhD in human genetics. We still have to go for programming classes, databases, and all that to know that how do we handle the data. So the future biotechnologist is not just playing with the DNA, RNA, and protein, but also doing programming. And that is where we have captured this in the form of the offering from our center. 2 years back, we launched the MTech program in synthetic biology, biomanufacturing. And as you can see, this is the need of the hour. It is not something that we have offered, which is away from the mainstream. But this is the need of the hour. Your biology is actually only 60 to 65%. The rest is chemistry and also the statistics, bioinformatics, AI and quantum computing we are offering from this semester, and entrepreneurship and so on. So this is the need of the hour. One has to catch up. If you don't catch up, as you can see the previous slide, there is this one. Yeah, there's an acute shortage of the skilled labor. So if you don't have the skill, the opportunity gets killed. So one has to be very, very fast and very prompt in being flexible in acquiring new skills. And many times people ask, are there any real life examples where synthetic biology has worked, writing the DNA or editing the DNA? And you can see there are live examples where it has worked. This is a classic example, which was back then. This was it made actually synthetic biology quite popular. This particular innovation at UC Berkeley. And then it was taken, and it was actually his postdocs, J. Kisling's postdocs, who came up with the new enzymes that were more efficient, and they spinned off the Amyris Biotechnologies. And Bill Gates and Melinda Foundation came up with, I think, more than $45 million. And then One World Health and Sanofi came in. So as you can see, there's a long journey, but it starts with this a dream of innovation and engineering biology for some large-scale application. The question is, why has engineering biology exploded now? Why not earlier? It's because very simple that your reading technologies have matured, your writing technologies, your editing technologies, and added to that artificial intelligence. All of them have matured now. And the big daddy who is coming very soon is the quantum computing. And this is something that everybody has to be aware that we have to understand what is quantum computing. We have to understand how to use the quantum computing tools, because now you have a power of seeing billions of combinations, not just thousands and hundreds, billions of combinations, and computationally screen them and see which ones are going to be working to generate stable variants. And that's where the future bioinnovation pipeline will have not only the bioinformatics, but also the quantum AI. Now, quantum will generate a lot of data, but it will not know that what's the direction to be taken. So that's how the AI comes into picture. It will give a directed evolution to the quantum data. And then because of the bioinformatics tools are currently being sharpened to this level and remove the errors and so on. And then instead of doing hundreds of experiments, you got to do only 2 or 3 and deliver the innovation at the market. And this was exemplified recently by Amravati Quantum Valley. So if you are interested in quantum computing, you must enroll for this course. It's an online course, but the first batch was 50,000 students. And I was told we have been given the responsibility of establishing the global quantum biofoundry there. And I was told that there's a queue of 5 lakh students in the queue for getting the quantum computing education. So it's time that we self-learn. So everybody cannot get enrolled and become expert very quickly. Of course, there are ways to avoid that queue, and you can always enroll in other courses. And then there is a self-learning material available on the net. So I would strongly encourage biotech students to get into this quantum space slowly, and then learn AI, and then you'll be a very, very strong biotechnologist. So essentially, if you look at the whole journey of synthetic biology, it is the synthesis of the parts, assembling into the genome, and triggering a metabolic or the regulatory event. And this was exemplified by companies who are now using organisms for large-scale manufacturing. And OPEX Biotechnology is using this. And you can see this is 20 to 30%, 50% cheaper, and phenomenal reduction in the greenhouse gas emissions. Likewise, the metabolics companies. Craig Venter took this to a totally different level. And this was a 10-year project, 400 crores, headed by a Nobel laureate. And they had this challenge of booting a cell with a chemically synthesized genome, and they achieved this. And it was a phenomenal, but of course it cannot be scaled up. So which means once somebody has shown the path, the next thing is to make it faster and affordable. Okay, that is the next generation of the scientists. They will involved in that. And we found that an evidence of that in terms of Visu, who was the first author on the paper, from John Hopkins PhD student. 5 years it took him to synthesize redesign chromosome. He made it lighter and he cut it into small pieces, and each piece was given to a chemical synthesis company, and they gave him these small bits and pieces of the DNA, and he assembled them and composed the entire chromosome in the yeast. And we found that it is not only lighter than earlier, but also it is functional. And then he put some watermarks on that. So this is a phenomenal science, which was actually covered by Craig Venter as an opinion piece on this article. So this is where the whole science is moving. Now we are also talking about the 6-base DNA. We're not talking about the 4-base DNA any longer. From 20 amino acids, the capability has increased to 172 amino acids. And that is where the innovation is, the front end of innovation is existing. So there is a prediction that by 2030, microbial strains will be ready for precision biomanufacturing. You have to quantum and AI enable the microbial strains. So you design the strains and compose them, and there you go. Well, Colossal Biosciences has taken this to a very different level, and they are now talking about de-extinction efforts to bring back the extinct animals. Where it takes us, we don't know, but there's a very interesting angle to this, and it has got certain concerns and certain proponents of this. However, in our lab, we have a simple, plain vanilla mission. We are looking at the dark matter of genome, which has remained unexplored. Today, we don't have time for that. We have already run out of time, another 2 minutes to go. So we'll be covering this in the next talk on Friday, August 7th, and we'll discuss in detail what is dark matter of genome and what are the various developments there and what are the various applications. So for today, I guess I hope you got a good start of understanding what is synthetic biology and what are its applications and what are the various aspects where industry is, trying to get a firm grip on this field. I like to stop here and I like to thank you very much for your kind attention, and I'll be happy to take questions. You can unmute yourself or you can write them in the box, the chat box. So thank you very much.
Hi, Pawan. Thank you very much for this wonderful talk and introducing the history of, the synthetic biology and how it developed into an industry scale and going into, biomanufacturing is really wonderful to understand. You were showing, the bio truth table, which is quite amazing. I mean, so I was just thinking, like, how can this table be designed to ensure, kind of a what do you say, reliable computation despite, because there can be fluctuations in the inducer or concentration or protein expression and environmental conditions. So despite that, how can we design this table to ensure this reliability in the output? Basically, I mean, that's one of the questions I was just having. It's very interesting to understand. Yeah.
Thank you. Thank you very much, Vinay. Yes, this was the wish list. It was
Yeah.
not never put into practice simply because initially, yes, they put into practice, and I'll touch upon that work also.
Yeah.
But people realize that because we're talking of analog system, we're essentially talking of a system where we do not have a control because it's
Mm.
nonlinear.
Yeah.
So
Yeah.
how do you compose a system where you have lack of control? Essentially, that's the inherent limitations, right? So yes, the truth table was essentially woven around the concept that if you have the inducer concentration on one side and the protein concentration on the other side, a box in the table will indicate the state of the system at that point in time. However, the state of the system also changes at every moment, and you have seen the gene expression fluctuations. So, how can you say that this table can be useful? So then people sort of retired this line of thought, and they said, okay, our bigger challenge of, in fact, enabling this table is let's make the analog to a digital version. If you can digitalize biology, then you can predict the designs. So then there was a 5-year effort by Driv Andy's students. There are two PhD students at MIT. They were given this task of creating a step function so that it is like um, you have gene is off and suddenly it is on. It is a straight line going up. There's no wavy stuff. And then this is the second state. They spent 5 years and they were barely able to show the step function. However, that step function was not stable, as you would expect. Yeah, because here we are talking of 4 billion years of intelligence, and what can a couple of weeks of experimental protocol do?
Yeah. Yeah.
So there are some deeper aspects which we cannot handle. So maybe on the wiser side, it is better to think of rational design and random mutagenesis and directed evolution, and then
Yeah.
selecting those designs, doing the large scale screening and finding the right designs in a soup. I think that's the best bet, in my view.
Yeah, with definitely with a multi-parametric analysis using the AI, agentic AI, and I think going forward may eventually answer this question, I believe. Yeah.
Yes, yes.
Thanks a lot, Paul.
Lot of
Thanks a lot.
thank
Yeah.
you. Thank you very much. Thank you very much. Oh, I think we have a question in the chat box.
Good
How
afternoon,
can
sir.
yeah, please. Good afternoon.
Actually, I am from MTech student from NCH. I wanted to ask that if I want to do synthetic biology for PhD, means how can we start? Means it is a new topic and too tough. So how can we propose a for interview proposal to for PhD interviews like that?
Yeah, we got to read the publications first. Read the last 5 papers, in my view, and they should be review papers because the review papers give you the story, opinion papers, commentary papers. Do not go for technical papers right now because we are not yet laid a firm grip on the concept. So start with the review paper, opinion, and commentary. Go with the YouTube tutorials, and then once you understand, it gets sort of absorbed what synthetic biology is in terms of the publications, not in terms of the concept, but in terms of the work that has happened so far. Then you go to the discussion portion of these original papers. So the second level is read the original papers, go to the discussion section, do not read the rest of the paper, just zoom into the discussion section. From there, you will get a number of unanswered questions. Extract them, make a list of them. Let's say you read 20 or 30 papers, not the whole paper, just the discussion portion. From there, you extract the questions, and then you see which ones make sense and which ones you can answer in the lab. And that becomes your raw material for the PhD topic. But it has to be done in
Thank you, sir.
stepwise. Don't rush. Yeah.
Okay, sir.
Okay.
Okay.
Thank you. Great. Thank you very much for this wonderful question. I'm sure a lot of people are, in the same way as to which topic will they want to work on. So yeah. So the question from Burhan is, how can synthetic biology aid in cancer research? Well, Burhan, the point is that synthetic biology is about designing parts, and these parts can be DNA sequences. These parts can be RNA or protein or peptides. And when you take, let's say, an existing peptide, okay, and it has a binding to a target. Now you have the AI tools, and now maybe in future the quantum computing tools that can sharpen the whole process. When I say sharpen, what I'm trying to say is that can achieve the docking of the therapeutic level that you need for a tighter binding event, and it can redesign molecules for better efficacy. So now, how do you do that? In the traditional setting, you mutate sequences randomly, or you have some idea about the protein structure and you say, okay, if I hit the core of the protein, I destabilize the whole stuff, or I look at the periphery, I mutate that, then what kind of binding pockets will appear or disappear? So you start, asking these kind of questions. However, when you can do those things in silico without actually doing experiments, because it takes months and months. I remember I was designing a transcription temperature sensitive mutant in Japan. It took us 6 months and we could not get the results, even though we had very good idea about which areas to mutate. So experiments can get pretty cumbersome. That's why please get into the computational aspect of designing biology in the computer, designing interactions in the computer. And then once you have a good docking score, and then you go to the MD simulation, the molecular dynamics simulation, you have a pretty good score. And then go and design it and test it against in the in vitro system, test it against. So there's a huge body of work around that. People have also used microbes to latch onto the cancer cells. That was also one of the works people were trying to do. I do not know current status of that. This is more than 10 years back. And likewise, our work on dark genome that you would see next time. This is all about therapeutic peptides. So we can make new molecules from sequences that have not been read in the nature. And likewise, people are creating new chemical entities. And so it's all about design. And it's all about once you design in the computer, then you go to the synthesis, and then you train yourself and learn, and then you deploy. So that is the reason why Biofoundry is very important, because it allows you rapid prototype testing. And a lot of work is available in that. So I'm sure you will come across a lot more. If you have any questions, I'll be happy to answer them after the talk. Yeah.
Yeah, Pawan, can I add a point here?
Please.
Yeah, okay. So adding to Buran's question, so already there are multiple antibodies. Synthetic antibodies are already developed, and FDA has approved those as therapeutic, strategy against oncology. For example, trastuzumab, which is against the HER2, and blocking or treating the breast cancer. And similarly, there are those cancers which are dependent on vascular endothelial growth factor based system. So there are synthetic antibodies which is designed and then approved. They're called anti-VEGF antibodies, which can treat VEGF induced cancer. So they're already in the approved FDA realm, actually. Yeah. Thank you.
Yes, Sunny, thank you very much for this add-on. Yeah, that's very important for them to know that it is not just conceptual, but it has already gone to the level of clinical trials. Yeah.
Clinical trials and approved.
Excellent.
Approved. Yeah. Thank you.
Absolutely. Yeah. So I guess people want a break. It has been a 55 minutes of non-stop lecture. So I guess
it's an interesting talk. Yeah.
So, well, thank you very much. Oh, I think there is
You have a question.
Question by Veena. How synthetic biology can help? If you want to unmute and speak, I think that's fine. Or if you're typing, I'll wait.
Hello.
Yes, Veena?
Yes, sir. Sir, I wanted to know like how this synthetic biology is helping for the de-extinction of woolly mammoth.
That's a bit of controversial,
Yeah.
area right now, and it has not been received favorably by the biodiversity people because, the concern is that, okay, you can do this woolly mammoth project by reconstructing the archival DNA that you get in a bad shape, and then you can fill in the missing pieces from the elephant DNA, and then reconstruct it computationally, and then go for the synthesis. The point is that what do you achieve by that? Because the ecosystems have been lost. If you go to the forest, there is hardly any food for humans, or the forest dwelling animals. There is hardly any food. So when you make something like this, then where are you getting the food from? People are now saying that, and the ecosystems are lost, the niches are lost. So, well, people are saying that let us then try the recently de-extinct, the recently extinct animals. Let us de-extinct them. But even then, the food chain should be available. Otherwise, it becomes pretty hard to sustain that momentum.
Yeah.
So it has not yet received, even if it would have been done in India, probably the clearance would not have been given. But then people are still playing around science, which is fine. As long as they don't any biohazard, it is fine to do science.
Yeah, correct. Thank you so much, sir.
Thank you. Thank you so much. Great question. Yeah. So there is another question from Biplab. How the synthetic, how synthetic biology can create a real mammoth traits. Yeah, the point is that when you have a standard parts, let's say an eagle can see a object, a mouse 5 kilometers away, right? We cannot see a mice 5 kilometers away at that level of resolution because we only see trees and mountains and a road. But an eagle can see that. So some people are saying, how about we transplant eagle's gene in humans so that our eyesight is much, much better? Well, a lot of such experiments are going on, but are we jumping the boundaries of ethics, or will it be stable? Now, you also hear that there are scientists in some countries who have made a commercial service of producing what we call as designer babies. So you select a menu and you say that I want a baby of this features, and they'll say, okay, we'll do some trials, and hopefully one of them will be successful. But that is little too hard. I think it's not good to step into this area. Um, there is excitement, intellectual excitement, of course, but whether we are crossing the line, I think we have to be very clear about that. Yeah. So it's in a way possible that you use computational tools to fill in the gaps, and you have an inventory of genes, and you know where to lift it and where to place it, copy paste genes, and then you ask the DNA synthesizer to basically create a synthetic genome. All those things are fine. That's not the challenging part. The challenging part is that when you go from DNA to an organism, you have to go through a lot of failures. And is it worth it? So, yeah, I guess we have run out of questions. So, I guess, shall we close it, Dr. Unikrishnan, or what would you?
Yeah, I think, I guess if there are no more questions, participants, thank you so much for attending this program. And we'll be looking forward you for the next event, which is on August 7, is it, Paul? Yeah.
That's right. Yeah. So that'll be the dark matter of genome. Well,
Yeah.
that is something that related to our work. Yeah.
Thank you so much for the participants, as well as, Professor Pawan.
Thank you. Thank you for this opportunity, and look forward to seeing you all again. Thank you.